Okay, so welcome to our last lecture before the midterm. Yes, I know. It's a nice way to get introduced for the day, which basically means that today we'll talk about the muscle lecture, of course, introduce the lab, and then do a little bit of a midterm review, not in terms of concepts, but more in terms of questions, what to expect, especially multiple choice and a little bit of the short answer. I apologize if I haven't got to your email. As you can hear maybe by my voice, I'm somewhat battling something, probably a virus. So in the next few days, I will be catching up on the emails, so not to worry. And the midterm we talked about, we'll review today. Okay, so today is muscle lab. We will start with the very basic skeletal muscle structure, the tendon, some naming terms that are going to be used, and then focus on the excitation, how do we excite the muscle, and then the molecular basis for its contraction from the perspective of the skeletal muscle. That will be the prototype. We'll talk about the molecular components, and then move on to a little bit of differentiating smooth muscle from skeletal muscle. I'll mention a few details on cardiac, but cardiac we have a whole 3 lectures dedicated to it, so we'll revisit it again. Last thing, we'll talk about muscle tension at rest, muscle force, tetanus, and finish off with a little bit about metabolism, what type of energy does the muscle actually use. Where this fits into the grand scheme of things is from a skeletal muscle perspective, this is your sports physiology, the ability to contract, the ability to do exercise, lift, balance is huge because you need to be able to adjust the right amount of tension. So from skeletal perspective, this is mostly day-to-day walking business. From a smooth muscle perspective, the smooth muscle, it lives within the blood vessels, so it will influence blood pressure regulation. It also lines the smooth muscle tract, the intestinal tract, so it's also contributing there. So smooth muscle has a lot to do with the cardiovascular section that we'll head to later, and with the next semester when we talk about digestion. So first off, here we have our basic muscle. So you have the belly of the muscle, or the meat of the muscle, where most of the contractile force resides. And the end of the muscle is attached to the bone through a connective tissue called the tendon. There's a little bit of elasticity within the tendon, so it allows for a little bit of stretch. But otherwise, it's a fairly dense connective structure that is there to provide kind of length, anchoring or leverage. So the muscle has something to attach to. The muscle is divided into a few components, kind of getting smaller and smaller. The outermost shell is called the epimysium, and that's the part that connects, kind of a connective tissue that surrounds everything, the entire muscle altogether. When we zoom in a little bit further, you can see that there are groups of muscle tissue called fascicles. So this is 1 fascicle, 2, 3, 4, 5 that are illustrated here. And each fascicle is surrounded by a perimysium. So also connective tissue, but specifically divides this portion from everything else. The next group is the muscle fiber that you see here. And the muscle fiber, this is now a cell that we're talking about, and it's protected by the endomysium. So we have epi, which is the most external, peri, which is in the middle, and the innermost layer that surrounds the individual muscle cell is the endomysium. Okay. So again, we have our fascicle here that is illustrated, and within that fascicle, this one long band is one muscle fiber. So the muscle fiber is the muscle cell. It's a multinucleated cell, and it has no communications with other muscle fibers, in the sense that it has the capacity to contract by itself. When it contracts, it doesn't cause a contraction to propagate to other units. It's also innervated by only one branch from an axon, from a motor neuron. So that means that you have the capacity to contract one single unit, one single cell at a time. That's a lot of precision, right? And that kind of precision will come into play as you're writing, the fingertips, minor fine finger movements, because you have this kind of precision control. So zooming in a little bit further. So this is our one muscle cell, so the muscle fiber. And within it, in the dots, you can see these are nuclei, so multinucleated. And within each of these cylinders, these are called myofibrils. So one muscle cell is composed of several myofibrils. And it's the myofibrils that are contractile units. So we'll be spending a lot of time zooming in on their structure and how they function. The muscle fiber is surrounded by a plasma membrane called the sarcolemma. And it's the sarcolemma that interacts with the motor neuron. So the neuron, last week you learned a lot about action potentials, depolarization, how they're generated. So the action potential travels down to motor neuron, and it excites the sarcolemma, which then propagates this action potential through the T-tubules, which are in blue, from the external portion down to the deep portions of the muscle fiber, where you have all the other myofibrils. So we have the T-tubules doing this, and the myofibril, when we look at its structure, we're kind of zooming in at each unit one at a time, and then we'll put it together in terms of how functionally they work. But when we zoom in on the myofibril, you can see that it's basically made up of two different strands, two different bands of proteins. And these two bands have the capacity to move away from each other or come closer. And the coming closer, that's their contraction. So on the one hand, we have the actin filaments. These are illustrated here, a thin little bubbles that are stuck together. That's the actin. And underneath, in the middle portion, this is the thick filament that is made up of a protein called myosin. So you have a long band of a protein with a head attached to the end. So that's what you're seeing here is the head of the myosin and then long bands that are extending from it. And on here we have the myosin in the middle, myosin, and on the outer portions, these are actin filaments that are all stuck together at the Z line. The linear protein that you can see on the actin that is kind of like a spiral going around it, this is the tropomyosin. And it doesn't have as much of a structure component. It's not contributing to contraction in the sense of giving it power. What it does is it helps to regulate whether contraction will actually take place or not. So today I'm really trying to slow down. So if at some point you get bored, please let me know. I want to hear that I need to speed up at some point. Okay. So we have one more protein within this kind of mess for now. And this last protein is called troponin. And it sits on top of it sits and interacts with a few different structures. That's why it has three subunits. So we have troponin I, which attaches to the actin. So this is where it's sitting on top of those bubbles. We also have troponin T, which is another component of the same protein that is attaching to the tropomyosin. So T for tropomyosin. And last is troponin C, which is sticking out, facing the cytoplasm, and it's waiting to be bound to calcium. So C for calcium. These subunits, by the way, are relevant not just to try to understand how contraction happens, but because we can actually test for them. In particular, troponin C, this is a test that could be ordered blood test to see if there was any muscle damage. More specifically, it's used to say if there was a heart attack that took place or not. And there are different genetic variations between the cardiac, the expression of a cardiac troponin C, and the expression of a skeletal muscle troponin C. So you could, if you have really good precision, to some degree of confidence say that this was a heart attack and it wasn't because the person was exercising and maybe damaged some of their muscles. Okay, so to review, so we have the myosin filaments. These were the thick filaments arranged in the center of what is called a sarcomere, which is one chunk of the myofibril. So if we go back just for a brief moment, so we have a myofibril, which is a very long kind of cylinder. And you can see in the black lines, illustrating we have one sarcomere attached to another sarcomere and another sarcomere. So you have them all linked together. So now if we look just at the one sarcomere, we have the myosin in the middle, representing the M line. And we have a Z line, which is used to anchor the actin. So you have actin facing towards one sarcomere, and then on the other side, there would be actin filaments projecting to the other direction. When contractions does take place, you have overlap between the actin and the myosin. And you will see that it's the myosin that moves relative to the actin. It's going to attach to the actin bubbles and kind of move them along, jump, attach to the next actin, move that one, push, and in doing so, it shortens the sarcomere and creates the overlap. Okay, there are two more structural proteins to introduce. First is titin, and where it sits is between the M line and the Z disc. The role of this, so you can see it as a spiral attaching the myosin filament to the Z line, which is another group of proteins. Its role is to make sure that as the muscle is being stretched, these two things don't separate completely. So in a way, it's helping to prevent a muscle tear or a muscle rip. To a degree, because we know that muscle tears do happen, so this isn't an all-powerful protein, but it does provide some elasticity so that you could stretch a muscle, but within limits, because you don't want these things to come undone completely. It so provides the scaffolding, it kind of keeps the M line anchored to the Z line, and it prevents the overstretch from happening. So you can see that, hence why it's kind of an elastic, so that as you stretch a little bit, it'll elongate, and then it'll fold it back into neutral position. So it's not contributing to a contraction force, it's more just bringing things back to resting phase. And we have one other protein. The last one is called desmin. So this is the best illustration that I could find, so it looks a little bit different. So in the middle, we have the M line. We have the myosin heads, and you can see that the heads are actually really well illustrated as projection as heads. In pink, this is the actin filaments, and on both sides, this is the Z line. So desmin attaches the Z line to the sarcolemma. So each individual sarcomere has a Z line on both ends, and then there's another sarcomere and another sarcomere. And we need to make sure that this contractile unit is somehow attached to the external sarcolemma, plasma membrane, that encapsulates this muscle fiber. Right? You don't want just internal things happening that have nothing to do with external shape. You want the entire thing to be able to contract and expand. And that's what the desmin provides. It anchors the contractile unit to the plasma membrane, the sarcolemma. We know that this is very important for muscle function because genetic disorders where desmin is mutated and it's not really functioning properly result in what is called as muscular dystrophy. There's many different muscle dystrophies. This is just one example. In that case, the person struggles with, to a various degree of movement. Walking will be difficult. They'll suffer from muscle weakness. It may be progressive, and it usually strikes early on in their childhood. Because basically every muscle contraction, although you're putting a lot of effort into actual contraction, it's not actually shortening the entire muscle. It's just one component of it. So the power that you're able to generate is significantly less. Okay, so this completes the structure component. So we talked about the actin, the myosin filaments, the troponin, tropomyosin, and two structural units, titin and desmin, and how they fit together. So now we'll move on towards the excitation component. So to come back, we have the sarcolemma, again, the plasma membrane that is encapsulating, surrounding the muscle fiber. within which you have several different muscle myofibrils, which are those contractile units. So now the first problem. The action potential that is going to come from the motor neuron is going to jump from the motor neuron to the sarcolemma. Okay, so the sarcolemma is this big thing on the outside. How do we get the action potential from the outside to the core? Sure, you'll have some myofibrils that are located externally, kind of on the periphery, but you need to get it into the core of the muscle fiber. And for that, you have the transverse tubules, T-tubules. So you can see that they're pretty much hugging the different myofibrils. And the action potential will essentially travel through the T-tubule. You'll have channels that are opening, allowing sodium to influx, and then like a domino effect, allowing the neighboring sodium channel to open and for it to influx. And that's how the action potential appears to travel. So it continues to propagate this way through the transverse tubules until it reaches each of the myofibrils. Once reaching the myofibril, now we need to go from the external environment of the myofibril to the inside portion. We do this through the dihydropyridine receptor. So DHPR, so this is the T-tubule. And in dark red, on the membrane, we have the DHPR, dihydropyridine receptor. Next to it is the ranodine receptor, which is sitting on top of a sarcoplasmic reticulum, which is holding on to the bulk of the calcium storage. So the point here is the action potential will travel, signal an opening in the DHPR, which will cause the ranodine receptors to open, and calcium will escape from the sarcoplasmic reticulum, where it can now initiate the muscle contraction. So to say that a little bit slower, once again, the DHPR is voltage gated. So it's waiting for that depolarization to come by in order to activate it. Once it's activated, it's actually physically complexed with the ranodine receptor, so that when the DHPR changes its conformation because it's been zapped by an action potential, it causes a pull, like a pulling action on the ranodine receptor, and that physically causes it to open up and allow for the calcium to efflux from the sarcoplasmic reticulum and flood the myofibril. So DHPR voltage gated, hence why we're waiting for that depolarization to come from the T-tubules, and it is physically linked with the ranodine receptor. This is very different, well, to some extent, from the cardiac muscle. In the heart, this whole experience is called calcium-induced calcium release. The reason for that is the DHPR in the heart is actually not connected physically to the ranodine receptor. They're two independent structures, and what the DHPR, it's still the same receptor though, what it does when it's hit with the action potential, it actually opens up and allows calcium to come in from the outside of the cell, and it's the presence of the calcium from outside that causes the ranodine to open, the ranodine receptor. Hence why it's called calcium-induced calcium release. We'll go over this again in the cardiac section, but I kind of want to prime you now so that you'll remember that there was something different about the heart in this particular arrangement. So again, so we have the question.
I was just wondering, did you say it was sodium that travels in the T-tubules, or?
It's an action potential that causes sodium, like, to influx through the channels within the T-tubules. Mm-hmm. So once again, so we have the action potential arrives at the DHPR, causes it to open because it's voltage gated, and the opening activation of the DHPR causes the ranodine receptor to open, calcium enters. So now we are at the calcium enters component.
Stimulates a muscle cell.
You think I talk fast? You'll have to watch this a few times. I apologize. This was, again, one of the better videos, but they talk very fast.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads through the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction. Age-contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber. Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump. Without calcium, myosin releases actin, and the filaments slide back to their original positions.
Okay, so we left off with calcium has just left the sarcoplasmic reticulum, and to slow down what was just explained, that calcium will bind to the troponin. And we had three components to troponin: troponin I, which was bound to actin, troponin T, which was bound to tropomyosin, and component C, troponin C, which is waiting for that calcium. So the calcium arrives, binds to the troponin, and that is what helps troponin to move tropomyosin away from the actin. The naked actin that remains is now able to bind to the myosin, which the myosin is kind of primed and ready to bind. As soon as the actin is available, it will now jump ship and kind of attach to the actin. So that's where we're going to start off now. And that's looking into the details of this interaction between myosin and actin. Sorry, I thought I saw a hand up. Or no? No? Okay. Okay, so again, before we start, the point of the contraction isn't that we're changing the length of actin or myosin, it's that we're helping them to overlap more and more and more. That's where the strength is coming from. So first off, at rest, we can see that the tropomyosin is blocking the relationship between the myosin and the actin. But the myosin is ready and waiting to attach to actin should the opportunity present itself. There's an ADP molecule, adenosine diphosphate, that is bound to the myosin head. There's also a small phosphate that is also bound at the same time. The phosphate and the ADP, we can almost kind of think of them together for this exercise. That they're there, but they're removed. So you have the ATP that was broken into ADP plus a phosphate on the side. And now calcium arrives. The calcium takes the tropomyosin out of the way with the help of troponin, and the myosin attaches to the actin. So that's position one. It attaches and it releases the ADP in the process, and it flexes. So attach, kind of flex, and ADP with the phosphate are released. Now we're actually stuck. So there was a movement that occurred. There was a little bit of a contraction. And if nothing else happens, these two will actually remain fixed kind of permanently. The actin and the myosin will be stuck. We see this when a patient, like when somebody dies and their muscles stiffen. It's because they're stuck in this form. They don't have any more ATP left, to move on to the next phase. So the next phase is very much dependent on the presence of a new ATP molecule. When the new ATP molecule arrives and binds to the myosin head, it allows the myosin to let go. And that's why in the case of somebody who, let's say, died, who passed away, their muscles go through a contractile attachment, kind of a reflex action, and then they're stuck there because the ATP runs out because there's no more life to generate the metabolism, and they will remain stuck in that situation. There may be other pathological situations where we are dependent on the ATP, and that may create problems if you don't have enough of it to allow the relaxation to occur. So anyway, so we have the flexion occurred, right? The actin, myosin, they bound, ADP left, we have the power flex, and now a new ATP molecule arrived, bound to the myosin head, and that allows the myosin to let go of the actin. Myosin will then hydrolyze the ATP into ADP. Myosin head has an enzyme, an ATPase, that has the capacity to do that. So it's not an external process. The head of the myosin does this by itself. So it breaks the ATP into another set of ADP plus a phosphate, and that is what allows it to not just let go of actin, but now go back to the initial position where it started from. So we've completed one cycle. We've attached, flexed, let go, and return to the status quo. As long as there's calcium present and as long as there's enough ATP present, we'll continue to do this. So if there's more calcium, great. We'll just let go of that actin, move on to the next one, and then move on to the next one. And in doing so, keep the sarcomere shortening and shortening and shortening. The calcium presence is dependent on the calcium ATPase. So there are a few of them throughout the cell, but two that are basic. There's one that is on the sarcoplasmic reticulum, so it helps to take the bulk of the calcium and put it back into storage, the sarcoplasmic reticulum. And there's another ATPase that pumps some of the calcium to the external environment and kicks it out of the muscle altogether. It's a smaller proportion, but it's still there. So you could play around with the calcium ATPase and say, what happens if this ATPase is working over time? It's working too hard. That means that the muscle contraction will be stopped prematurely. We wanted it to continue. We wanted to build up a stronger contraction, but because the ATPase was working really well, it actually depleted the stores, put the calcium back into storage, and now we're stuck. That's one way to make the argument. We can play it the other way and say that the calcium ATPase isn't functioning properly, and it's allowing the calcium to stay in the cytoplasm too long. So the calcium allows the troponin to move the tropomyosin, so it's allowing the muscle contraction to continue and continue and continue. In which case, as long as ATP is there, this muscle contraction would keep going and would probably result in a fairly kind of spastic position where you just can't really relax. These are extremes. I just want us to kind of gauge what the ATPase is doing. Yes?
Sorry, can you just repeat what happens when the pump is too active?
Mm-hmm. So if it's too active, right, it moves the calcium back into storage, which means that we would have to get another action potential to come by and re-stimulate the opening of the channels. So if that wasn't the message coming from the brain, then we would have actually shortened the length of the muscle contraction by removing the calcium prematurely. And another summary.
Contraction of a muscle. During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
Okay, I guess that was it. Sorry, I thought something else to any questions on this? While we have the video, this is actually a good illustration.
So this is happening several times over the course of just one contraction.
Yes and no. So we'll get to a point where we're talking about muscle twitches. I'm just going to fast forward to answer your question. So when you have, this is what a nerve sends out, this is what the muscle is doing. For the one single action potential, how many sarcomeres, or rather how many of the cross bridges will be formed? Maybe it's one, maybe it's a little bit more, maybe it'll be 3, but it'll be a very small amount for one muscle, for one action potential coming from the brain that is sending that signal. It'll be a fairly small number of cross bridges that will be formed. I'm not 100% sure if it's just purely 1 or if it's like 3. But in order to get a contraction going that's strong enough to lift, let's say, a cup, you would actually need to get several action potentials to be sent back to back in order to get not just a few cross bridges, but like to get multiple cross bridges to occur, and not in just one sarcomere, but maybe in, let's say, 50% of the myofibril, and maybe not just one myofibril, but like 20 of them. And it'll depend on what it is that you're trying to do. This isn't too heavy, so it's not going to take that much. But if you're trying to lift something that's very difficult, it would require a lot of nerve signals to go through in order to recruit multiple myofibrils, muscle fibers, to kind of say like, oh, you know what, all hands on deck, everybody contract. Does that answer your question?
Yes, and all of that happens within like split seconds because it's a voluntary decision that we make when we want to.
Yeah, exactly. Thank you for bringing it back to to a bigger context, because that's exactly it. It's quite fascinating that all of this is so fast. It almost seems instantaneous when it happens. Yeah. To add to this, though, the size is very small. So each sarcomere is 2 micrometers. So it's each sarcomere is a very small unit. So those cross bridges that are taking place, they're already kind of close together. So you're shrinking kind of by 1 micrometer each of the sarcomeres. Maybe that's where the instantaneous part is coming in, because to travel that tiny distance for 1 sarcomere isn't that difficult. The fact is that you're sending a signal that's going to 1,000 sarcomeres at the same time. And so each of those is shrinking just by a little bit. But it's the cumulative effect that creates that contraction. Any other questions on this before we move on to?
I have a weird question, but if someone, let's say a bigger person and like long muscles, they're going to require a higher amount of ATP, like within their body to
power
contraction of somebody that's smaller.
Yeah. Not necessarily to do the same contraction, but the person who has bigger or longer muscle would be able to maximally lift something that another person wouldn't be able to. And to do that, they would need the extra ATP. So if we're both lifting the same cup, we'll use the same amount of ATP because we'll be using the same number of sarcomeres. I'm averaging though, I'm not 100% on that either. Just because the weight of this is going to be the same. So tall, like longer muscle means that you can create a more power for the muscle, so you can lift heavier. But if you were lifting the exact same thing, I think you would use fewer sarcomeres because why would you? It's very light. So you and me would probably decide to use the same number of sarcomeres, or let's say that you're very tall and I'm very short. I would still use 5 to lift this, and you would also use the same 5. There would be more of them. Oh. Yes, exactly. But to achieve the same weight, I believe that we would need the same number. Not 100% sure on that, though. I want to check into that. Does anybody know, actually? Or has done the math or has looked into this? Would be in like sports med, I imagine. The clinics that are going to look at like the best athlete and try to figure out how to get the edge. No? Okay. I'll look into it. Short versus ATP. Okay, so now then we'll move on towards the smooth muscle. And the smooth muscle has a few major differences. So unlike cardiac muscle, which actually looks fairly similar to skeletal, the smooth muscle is quite different. So to start with, you can see that it's not this nice, crisp line, linear cell. It's actually a little bit more of a blob. That's one. Second is each cell has only one nucleus. So we have a very clear, this is one cell. A few more changes. We have gap junction. So we now have a way where one cell can communicate with another cell. So if I contracted, and I think that it would be helpful for my body to contract, I'll send the action potential in their direction through the gap junction. If you recall the gap junction, it had the 6 connexins on both sides. So there were 6 connexins that were arranged to create a pore on the cell A side, and then there were 6 connexins that were built into the plasma membrane of the nearby cell. And those 2 connexins would kind of conjoin so that you create a pore. So you now have a gate, a tunnel, through which you can send ions from one cell to the other. So if I'm undergoing depolarization and I have more sodium channels, and I think, great, let's all contract at the same time, those sodium channels, those sodium ions will flow through the gap junctions to my nearby partner from cell A to cell B. And in doing so, I will now spread the depolarization to them, and they will now start to contract. So unlike skeletal muscle, which are waiting for the nerve impulse to come down through the axon in order for cell A to contract and cell B to contract, smooth muscles can talk to each other, so they can influence each other's contraction, which means that they work more as a unit, as opposed to independent skeletal muscles that are waiting for the brain to create the teamwork environment. The smooth muscles can kind of organize themselves into a team. A few more differences. So if we take this smooth muscle here, and we'll kind of zoom in on a little bit of what's happening in the structure. There are dense bodies that are used to anchor the actin filaments. So where is our myosin? So here we have the example. This is the myosin, what was the M line before. And we have actins that are attaching to a round ball, the dense body. And so here, dense body, myosin, and then actin filaments around it. So you can see that, again, there is a little bit of a linear component to it, but there's other directions now. The skeletal muscle just had 2. We were either kind of stretching or contracting, but it was a very clear line. Now we have a little bit more 3D. We can kind of contract inwards, expand in different directions. So that also adds to some complexity. You can see here a contracted cell that is definitely doing a lot more than just one line contraction. So lots of differences in the structure. The gap junctions the dense bodies, the single nucleus, the single cell talking to a friend. Those are the main ones. Yeah.
So for skeletal muscle, the muscle fibers go from tendon to tendon?
Mm-hmm.
Oh.
Yeah, exactly. And so their contraction is pretty much that direction. You have a different, why we can do more than just one action is because you have other fibers that are running in other directions. But that one muscle is only contracting in that one direction. You can orient it, put it on an angle, but it'll still do the same in or out. Versus this is contracting with just that one cell is contracting top, bottom, front, and back. So it has more versatility. Mm-hmm. Yep. So the dense body is kind of like the Z line. It's providing an anchorage from where the actin filaments project from, so that as contraction takes place, you'll have two dense bodies that are coming closer together. And they're also attached to the external part of the cell. So they'll be attached to the cell membrane, so that as the actin moves closer, the entire cell is being pulled to contraction as well. So the internal portion is also different. We do need calcium, so that part stays the same. The calcium is coming through from the sarcoplasmic reticulum. We now have two channels that can deliver it. The ranitidine receptor is still there, and when it opens, calcium leaves. But we also have another receptor, which is called the IP3 receptor. So we have two ways of getting the calcium out of the sarcoplasmic reticulum. And we also have different ways of getting this to work to begin with. We have voltage-gated, and we have ligand-gated channels. So now we're not just waiting like with the DHPR receptor, which is just voltage dependent. We have this voltage dependency, but we also have ligand, which means that we're waiting for either a hormone or a neurotransmitter to arrive to trigger the contraction of the smooth muscle. This is very useful because those ligands, this will play into the contraction of the smooth muscles within the intestines when acetylcholine neurotransmitter will stimulate this contraction to occur. So you have a way for the smooth muscles of the intestines to generate their own rhythm. So they actually periodically contract on their own. That's voltage dependent. And you have another mechanism where it's the acetylcholine that is coming from the brain that can say, okay, now would be a good time to contract. You have your rhythm, but for whatever reason, we need you to speed up the contraction. We see something that you don't. So we have more variety, more options with the smooth muscle. The next part is the fact that we don't have troponin. So troponin was our gatekeeper. It was what was allowing us to regulate whether we contract or we don't. Was because troponin held the tropomyosin, could pull it out of the way, and when there was no calcium, it would move it back. Now we don't have troponin, so we need another gatekeeper to say when the smooth muscle contracts and when it doesn't. In this case, the gatekeeper is the myosin itself. So myosin, when it's not phosphorylated in the smooth muscle, it looks something like this. And this is not 100% accurate, this is more of a schematic. But it's, you can see that the myosin has a fine, but the long component is not. So this portion wouldn't really be able to interact with much with actin. Unlike when it's phosphorylated. Then we actually have a properly elongated myosin with the heads at the top. So this phosphorylation is the key to getting myosin to interact with actin and to allow the contraction to occur in the case of smooth muscle. So now we'll ask, well, how does this happen? What does it depend on? It still depends on calcium. And the pathway or the story is as follows. Calcium is released from the sarcoplasmic reticulum, either through the ranitidine receptor or through the IP3 receptor. It binds to calmodulin. Calmodulin then activates an enzyme called myosin light chain kinase. Kinase enzymes as a group are known for the fact that they will phosphorylate something. In this case, the something is the myosin. hence the name myosin light chain kinase. So it will phosphorylate the myosin light chains. And in doing so, it now activated the myosin, and now the myosin is able to bind to the actin, very similar to what we saw with the skeletal muscle. We have the go signal, so now it will bind, flex, bind to the next actin, flex, and strengthen the contraction. So once again, we have calcium. Calcium binds to calmodulin. Calmodulin activates the myosin light chain kinase, myosin MLCK, which then phosphorylates the myosin head. And the myosin light chains, and the myosin, now that it's phosphorylated, will actually interact with actin and cause the contraction to occur. So we have a lot of differences. The last difference is the least understood difference, and that's the relaxation part. We're still dependent on calcium leaving the scene. So we still need to get calcium out of the cytoplasm and back into the sarcoplasmic reticulum. That's a requirement. But we know that there's something else that happens, because even when you take all the calcium out, the contraction is still actually in this phase of contraction, the smooth muscle. It takes the smooth muscle a longer time to fully go into relaxation. Unlike skeletal muscle, if you take the calcium out, sure, that's it, we're done. There's no more relationship between the myosin and the actin. Here we're dependent on the calcium leaving, but we're also dependent on myosin being dephosphorylated. And that is done by the phosphatase. So again, very long name, myosin light chain phosphatase, which takes the phosphate off. And now that the phosphate is removed, okay, the actin and the myosin can't interact appropriately anymore, and they will detach. The activation of the myosin light chain phosphatase is the portion that we don't really fully understand. So it's well regulated, but there's a lot of different players in place, and that's why this is kind of beyond our course, but it is still poorly understood. So there isn't a very well-developed model where we can say A, B, C, D, and that's how myosin phosphatase would be activated. So for our purposes, relaxation depends on calcium being removed and on the phosphatase being activated so that it can dephosphorylate the myosin. Okay, we're almost done with the smooth muscle, and then we can break before we attack the skeletal again. There's another, where this all fits together, is that the ATPase within the myosin, which will still be required. The myosin head still has an ATPase because we still need the ADP in order to bind to the actin. We still need to get the ATP so that we can relax. So that internal rotation of myosin binding to actin, moving it, attaching to the next actin, is still very much alive. So that part is preserved with the smooth muscle. This part is very slow compared to skeletal. The myosin is slower to attach, and the myosin looks like it's slower to detach. The fact that it's slower to detach is actually really important because it means that the contraction phase is prolonged. And this is key because a lot of our smooth muscles are tonically activated, which means that you have blood that is pressing on the blood vessels. Those blood vessels are exerting a little bit of a contraction back. If this cost you a lot of ATP, we would probably cease to exist because we would run out of energy because constantly your blood vessels are in a state of contraction. Thankfully, that doesn't cost a lot of ATP. So this low energy requirement is a really important feature of smooth muscle cells. So we have, yeah?
Sorry, can you just repeat that?
Yeah.
Why it's good to have a slow detachment?
Yeah, absolutely. The slow detachment means that you don't need to use as much ATP. Everything is slow. It's slow to attach, but then once you attach, you're fulfilling a contraction, and you can stay like this actually for a very long time. Hours, I think it was 2 days. We think it's because the ATPase is working slower, and we think it's because it's slower to attach and slower to detach, and this is why the ATP is preserved. This is a hypothesis, so it may be true, it may not be. But having very low energy usage for smooth muscles, as I was saying, is absolutely crucial, so that your blood vessels can be powered at minimal cost. The intestinal cells, same thing. Those are constantly in a state of contraction, relaxation, and the eye, etc. Sorry, I don't know if that's a stretch. Was that a stretch? Okay. Yes, so low energy is crucial. And the last part about smooth muscle cells that makes them unique and very useful is this force tension relationship. When you stretch a skeletal muscle, it is actually designed to contract in response. It may be that when you act, there's many different actually explanations for this, but those explanations are not important right now. So I won't get ahead because that's the next semester. But with skeletal muscles, what we know is that as you stretch a skeletal muscle, it responds by contracting to keep itself together. Smooth muscle cells also do this initially. So as you pour more urine into the bladder, right, it extends. So you're stretching the walls of the bladder. There is a reflex to contract, but it's a very short-lived reflex. And after that, the muscle actually relaxes and says, yeah, okay, there's more urine here, whatever. We don't need to do anything about it. To a point. So you can keep stretching out the smooth muscle. It'll have initial reflex of contracting, and then it relaxes, and it'll relax around whatever it is that you stretched it with. So it might be urine in the bladder, it might be blood in the blood vessel. And this blood, by the way, in the blood vessel is what is considered very healthy. For us to be able to, for our blood vessels, to have the flexibility to relax around the extra blood. And it's a loss of flexibility that contributes to high blood pressure. When there's more blood that fills the vessel, and the muscle responds with contraction and stays there. That's a problem. We need it to be able to relax. So this is a healthy feature of smooth muscles, that in response to stretch, there's a very small reflex contraction that is followed by a relaxation in order to accommodate the tension. And where we see this clinically is with the bladder, where you'll continue to fill it. There's stretch, little contraction, then relaxation. Stretch, small contraction, relaxation, up to a point where eventually this is too dangerous and there will be a contraction to void the bladder. This seems like a very reasonable time to do a break. We may get booted off again, but if that happens, we'll restart. Okay, so now we've completed the story about smooth muscle and why it is so unique. And now we are moving towards just a very brief comparison, and then focusing a little bit back on the skeletal muscle. So for comparison, we have skeletal, cardiac, and smooth muscle, and we're comparing various features, whether it is striated, how do we control it, the location, is it getting innervated, and does it have troponin? So you can see a lot of similarities between skeletal and cardiac. They're both striated. They both have troponin. The skeletal is attached to bone. The cardiac is located in the heart. And the cardiac is not involuntary. So in the sense that you can't generally consciously increase the strength of your heart contraction. You can, to some level, change your heart rate. That we have experienced with biofeedback. If you check your heart rate, over time you'll actually start to slow it down. But the strength of the heart contraction, you got nothing, at least as far as we know. And it doesn't need to be innervated. So the cardiac muscle works independently of the nervous system. The nervous system can modify it, but otherwise the heart will continue to beat on its own even if you remove it from the rest of the body. Versus the skeletal muscle. It's mostly voluntary, although when you have reflexes, those are involuntary. So like the patellar reflex, involuntary to an extent. And we also have involuntary reflexes such as When you overstretch a muscle, it contracts. You don't control that. That's also an involuntary contraction. If the patellar muscle contraction, you can kind of override and grind your teeth and hold on to it and override it. You can't override a tendon relaxing. So some of them are involuntary. And this is a very important part, is if you damage the nervous tissue, the nerve connecting to the muscle will actually die. So it's not just that, sure, you have no way of accessing it, and so you can't contract it, but because the nerve has been damaged, the muscle will actually atrophy over time. And atrophy means you're losing those muscle cells. So this is kind of quite important from a rehabilitation perspective, right? As in, if somebody had spinal nerve damage, they may repair that damage. We know that those nerves do heal, but it depends on the time and what kind of damage will have happened to the muscle that didn't receive any stimulation. We see this a lot with muscle, kind of like you use it. If you don't use it, you lose it. Question?
What about for like anorexia nervosa? If you get muscle atrophy due to malnutrition, can that be gained back with clinical interventions?
Yes. So you can. That's a very good point. You can regain to an extent muscle that has been lost due to atrophy. Yes. There are outside of this, just off topic, there are satellite cells that are kind of like stem cells for the muscle, so you can stimulate new growth. Yeah. With a loss of a nerve to that muscle, it'll be a challenging scenario because now you have to grow both things somehow. Get the nerve to extend, repair, and regrow a new muscle. Versus smooth muscle, looks very different. Not striated, it's you don't have control on it, so it's kind of like the cardiac. It sits inside tubular organs like blood vessels, urinary tract, digestive tract, and also like the heart, it doesn't need to be innervated because the smooth muscle has the capacity to, or many of them have the capacity to self-generate a signal. Not all, but the digestive tract, for example, does very much so. So it continues to create an unstable plasma membrane that continues to depolarize and then come back, repolarize, depolarize, come back. It never kind of sits still. The way a skeletal muscle will just hang and wait until it receives a new stimulation. There's also no troponin, as we found out, and that's why we needed those two enzymes, the kinase and the phosphatase, to stimulate the myosin to be phosphorylated so it could contract. Okay, so now we'll zoom in on the skeletal muscle and talk a little bit about the strength contraction and how to get a more powerful contraction. So at the top, we have one single action potential that is being delivered by the motor neuron. Yes, last week we talked about the absolute refractory period, where once you've depolarized the neuron and you've opened the sodium channels, you've now kind of successfully entered the absolute refractory period because you won't be able to send another signal through until those sodium channels close and until they reset and are ready to be opened again. So that was the absolute refractory period dependent on the sodium channels. The muscle is illustrated below. So we have the action potential arrives. Soon after, the muscle calcium is released, binds to troponin, myosin and actin interact, and contraction begins. And there's overlap between myosin and actin, cross bridges are formed, and the sarcomere shortens. And then the muscle relaxes, and they detach, and the muscle goes back to neutral. If you send another twitch, and then another twitch, you will successfully not just prolong, you're not going to keep status quo. So you're not just going to get a nice plateau at that point. You're actually going to start to see summation, where we got to this point in our graph, and now you're sending another action potential, so we'll see another bubble kind of spike. And then if there's another action potential coming through, there will be another hit. So the strength of that contraction will actually build and build and build, because with each twitch, not only are you not allowing the muscle to relax, you're recruiting more of the sarcomeres, you're recruiting more of the myofibrils, more of the muscle fibers. So you're starting to kind of recruit more and more with each twitch if it's added. So if it's coming at a relatively close period in time. And this is where you can get to what is known as tetanus. So that is seen here. So we have one contraction, relaxation, contraction, relaxation. And then we start to see contraction, relaxation, starting to happen closer and closer together. And you notice that even though the muscle went back to relaxation, it never actually fully got a chance to relax. So those cross bridges came together, and they went maybe back a little bit, but they never got the opportunity to fully move apart to neutral position before another hit came. So they were forced to come closer together, and then another hit. And so this would be termed tetanus, where there is no break between contraction. There is no relaxation. There is just contraction, 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 where you've maximally engaged those cross bridges, and now you can't really, there is a plateau effect at this point, so you can't go any further because you've maximally engaged all the overlap between the actin and the myosin. And the term tetanus, you would have heard probably more so in a negative connotation, because we have a tetanus vaccine, which is given to prevent death by paralysis because you're in such a spasm. So in that case, tetanus that we use a vaccine for is used to target a tetanus toxin, which blocks the muscles from relaxing because it blocks the neurons. So the end result is the same, where the muscles are in a maximum in a maximum state of contraction, but unlike in a physiological state where this is purposeful, it's because I'm trying to lift something and hold it. Now I will be engaging in tetanus. I don't want to be relaxing. Then I'm going to drop what I'm holding. So there's a point where this is useful. But tetanus in a pathological state, when it's due to a toxin, that is lethal because it's happening to all muscles all at the same time. And why the person actually dies is because their respiration is paralyzed. In order to breathe, you're constantly contracting and relaxing, contracting and relaxing. So if you can't relax for your breathing, no more air is going to come in. So just to compare those terms, because like at the very end of the day, tetanus in both cases, it's the same. It looks like this. It's just that we don't do this action in all muscles all the time. You do it in a very purposeful way, probably in a few subsets of muscles where you're trying to do a very specific movement for a little bit of time. But if you were going to do it systemically everywhere, your front muscles, your back muscles, your triceps, your biceps, one is that's useless because they're opposing muscles. Two, that will be quite painful because they're opposing each other. And three, again, it's the respiration that's going to be really the problem. Okay, so this is, we talked about tetanus. So just some words, but this is pretty much what we talked about. And the other concept is the muscle length and optimum tension. So there's two extremes. The extremes are easier to work with. So one extreme is illustrated at position D. So this is the length of the sarcomere, and this is the maximum tension that it can produce. And D is illustrated here at the top. So what you can see is, in red is illustrated, this is the myosin, and then in black, this is the actin. So you can see that there is absolutely no interaction between actin and myosin. They're so far apart from each other that even if there's a variety, a ton of calcium that is dumped into the system, nothing's going to happen because the myosin can't see the actin. So they can't actually engage. And that's why position D, the sarcomere is very long. It's been extended, but there's pretty much no tension because there's no interaction between the actin and the myosin. On the flip side, so one extreme is you have an overly stretched out muscle. Maybe those titin proteins aren't functioning. Maybe this is another genetic mutation. Let's use that as an example. Versus A. So position A, now it's very short, in terms of the length of the sarcomere. We do have some tension, but it's not really optimal. And if we look at A, you'll see why. So we have, again, actin and actin on the bottom. You have myosin on top. And you can see that the actin are actually kind of smushed together. There isn't going to be effective, you've kind of lost it at this point. There was value when the actin and the myosin were interacting and the heads were bound. But once you start to smush the actins together because they're too close, you're also weakening the muscle. You're not completely destroying it. That's why there is still significant 80% tension remains. But the more you continue to smush it together, and now the next step, the myosins are going to start to collide with the myosins of the next nearby sarcomere. Now you're starting to lose force very quickly. Question?
Would the tintin kind of like that too?
The tintin in which scenario, A or B?
A.
In scenario A, so hold on, the tintin was holding the M protein, the myosin filaments, and attaching it to the Z line. So in this case, I guess the tintin would be completely squished to non-existence.
Would it be affected in that situation though?
I think it would be possibly damaged, but I don't think it has the capacity to prevent this from happening. As far as I know, its functionality is to prevent the overstretch, but it doesn't help to prevent the opposite, like the too much compression. Okay, and then we have the optimum lengths, which is B and C, where you can observe that there is good overlap between actin and myosin. We are forming cross bridges, and we're reaching maximum potential. We don't have that much more actually space. So B reached maximum potential. There's no more closer that it can bring those actin filaments without them starting to collide. So this is as good as it's going to get, which is why these are illustrated at 100% maximum tension. For skeletal muscles, this explains why if you kind of overstretch your muscle or maybe tear it, you start to see weakness. Where we see this model very kind of clinically relevant and quite important is with the heart. Because as the heart is damaged over time, if it's being stretched out because you have a lot of fluid that is building in the blood vessels, maybe blood pressure, maybe the kidneys are failing and you have a lot of fluid that the heart has to stretch around. If it is being overstretched too much, it ends up being in position D. And that's where its functionality starts to tank very quickly and it becomes very weak and failing, essentially. So this concept we'll see again when we talk about cardiovascular. We'll revisit it one more time. Yeah?
Could A be like a knot in your muscle? Is that the scenario that you see with A?
Sorry, can you, maybe I missed the beginning. Can A be?
A knot in your muscle. I don't know how to say.
A muscle knot. I'm not sure what that would be actually in this model.
Because a knot in a muscle doesn't contract like the rest of the muscle and has problems overextending.
That's very true. And it is like it doesn't contribute. So from a muscle strength perspective, yeah, maybe that's what that one is. I hesitate to say 100%, but what is a muscle knot? Could it be position A? Again, if anybody knows, please feel free to jump in. All right. So before we get to energy, just one final idea. And the idea is that there is also elastic tissue. So we focus a lot about the contraction of the muscle, but there is elasticity in the muscle as well. Some of that is between the titin molecule, for example, the titin protein is allowing a little bit of a stretch. So there is some elasticity between the sarcomere. So as one sarcomere contracts, there is a little bit of lengthening in another sarcomere, unless it also contracts. So the muscle cells themselves have that elastic capacity. And then we have elasticity within the tendons. So this is a very maybe helpful concept. In the lab, you'll talk about two types of muscle contractions. One is called isotonic, and the other one is isometric. So isometric is where there is no movement. So iso means stays the same, metric means length or the size. So isometric, you're contracting, but you're not actually seeing anything move. So for example, as I'm holding onto the cup, it's in the same position. There is a muscle contraction, but the cup isn't being moved up and down. So you can't actually see the contraction of the muscle. It doesn't look like it's shortened. You'd be able to see if it's shortened if I'm starting from this position, it's long, and then I brought it up, there's a little bit of a shortening that's happening here. So if I'm moving it and you can see it because of the contraction, this is isotonic. And if I'm not, if I just hold it like this, there's still contraction happening. This would be the isometric. So you'll go through this again in the lab, but I'll just give you a hint that this is going to become very important. And if you're walking out of the lab or you're closer to the end and you still don't see how this applies, that means that something happened and something's missing. In that situation, please feel free to post on the forum. Be like, I'm not really sure how elastic fibers relates to this, but I'm really hoping that this will help give a hint into the direction. Okay, so we are now approaching the last phase of today, and that's to talk about energy metabolism and how does the muscle sustain itself. So muscle has a way to store fast energy, ATP, in the format of phosphoryl creatine. Sorry, phosphoryl creatine, CRP. This is a very fast form of storage in the sense that it doesn't take a lot of time to create it. It doesn't take a lot of time to break it. So that if you're going to suddenly start to run, you forgot you needed to be somewhere and you dash out of the room, you're going to be using your phosphocreatine in those initial seconds. Before the rest of your body had a chance to accommodate the sudden need for using of energy, for the sudden need to do extra muscle contractions, you'll rely on this really fast fuel. There's only so much of it that you can store inside the muscle though, so this will burn out relatively soon, and then you'll need to actually use something more substantial, which is your lipids and your carbohydrates. So in general, and this is not 100%, but if you are going to do exercise for a while, then depending on what type of exercise you're doing, you may be using more of your fats versus more of your carbohydrates. If you're doing a long-term run, let's say, and you're using only up to 80% of your maximum capacity, sorry, 60% of max intensity, you will use the lipids. Why? Because you have enough time to actually access them. Lipids are amazing in the sense that they pack a lot of calories, a lot of energy is stored in a lipid, but they're kind of crappy in the sense that it takes forever to get that energy out. The biochemical reactions just take longer. So if you're going to do something long-term, like a run that doesn't require all the energy like all at once, you have enough time to get those biochemical pathways to work, to get the energy out. But if you're doing something that's super high intensity, I don't know, heavy weight lifting at a really fast pace, and you're doing it at greater than 60% basically of what you can do, you're going to be using carbohydrates because they're faster fuel. So you can get to them much faster because your muscle requires this fast energy to do this crazy amount of intensity right now. So that brings us to the next question. And the question is, or not the question, but the challenge. And the challenge is that eventually, oh, sorry, was that a question? No, okay. You guys keep throwing me off. Okay. Not so much a question, but just a comment that you make energy whether you have access to oxygen or not, up to a point. So we have aerobic pathways, and we also have anaerobic pathways, which produce some energy, not as amazing as aerobic, which is why we choose to breathe. But when push comes to shove, you still can make some energy without the oxygen, but then you're also making lactic acid, right? And that's kind of useful to let you know that this is problematic, so that you don't only feel fatigue because you're not getting as much energy, but also the lactic acid is quite unpleasant when it builds up in the muscle, hence causing pain. So this is why, essentially, as you start to run, let's put this into all together. You suddenly remembered there was an appointment, or you suddenly remembered you need to be running for whatever reason out of the class, and you bolt out of here. In the initial phase of that, your muscles are going through a lot of contraction, which a few seconds ago, they were just relaxing and not having to be engaged. Suddenly, so let's say 20 units of your muscles are contracting and burning ATP at a really fast pace. Okay, where is that ATP going to come from? Well, you have your phosphocreatine, so you can break that down, your emergency storage, and then some of your carbohydrates. But your air capacity, you didn't tell your lungs you're going to bolt, so there was no need for them to start breathing faster to get you the extra oxygen. You have enough oxygen to sustain you for what you're currently doing, which is sitting down. But if you suddenly bolt, it's going to take a little bit of time for the respiratory and the cardiovascular system to catch up to that. So initially, your muscles are deprived of oxygen. The rest of your body is going to call dibs on that oxygen. Your brain, the inner organs, that oxygen amount was calibrated for you sitting, so that, and the amount of oxygen needed to go to the brain to feed the liver, etc. If you suddenly hold, that's nice for the muscles that they want to use that oxygen. Your brain's not going to let it go. So that means that in those initial moments, you're going to be using anaerobic glycolysis, and you're going to be using that phosphocreatinine and whatever means you can to power yourself for the bolt. Eventually, within a few seconds to minutes, your respiratory rate will go up. And that's what we're seeing here, that your ventilation rate will increase in a kind of jagged way over time as your body figures out what it is that you're doing. By how much do we need to increase? So we increased quite a lot. Clearly, it's still not enough, so we need to keep increasing the amount of ventilation. And eventually, we meet status quo. That means that the amount of oxygen that is being ventilated, taken in, is actually proportionate to what your brain needs, your liver, all your organs, plus your muscles. Eventually, you're going to stop. You reach wherever you wanted to go. And at that point, you'll notice that although there is a drop in respiration, you don't stop, you don't go back to resting. And that's because you need to pay off and recreate that phosphocreatinine, which you were using and relying on heavily in the beginning. You never had time to make up for that in here. Because as you were running and exercising, your lungs were focused was on just, let's meet the demand right now. We'll worry about filling up stores later. Now that you've stopped exercising, this line represents you stopped. Now we can worry about replenishing your stores, getting rid of that lactic acid that built up. So keep breathing out quite a bit, expiring it out. It might not make sense right now, but the lactic acid, you'll convert it in a way that you can expire the carbon dioxide, and that will reduce your acidity. So that's going on in this portion. And you need to make up for the lack of phosphocreatinine, which you burned up really quickly in the beginning, and now you can phosphorylate it again by the use of oxygen. So this is called oxygen debt. So in the beginning, you were running really quickly. You created this debt because although your muscles did do the work and took the energy, there wasn't enough oxygen in the system to actually pay for this energy. So that's why you end up with an oxygen debt. Once you stop exercising, you actually keep breathing rather fast for a while in order to pay off this oxygen debt. Keep the oxygen coming so that you can continue the oxidative aerobic glycolysis so that you can power up and restore the The last portion, so we talked about the fact that you'll need to regenerate. The anaerobic kind of gives you the lactate. It gives you very little amount of energy. That's why you really want to get your ventilation out there so that you can use aerobic glycolysis and beta oxidation for those lipids. So where this ends is that you end up feeling tired at some point. And generally why that is more so because of the muscle not being able to metabolize the energy the way it needs to. So the cardiovascular system might contribute to this, so there's only so much that your heart can pump. The breathing system is actually rarely a problem unless you're not healthy, and then there's something obstructing your amount of respiration that you can do. But for the most part, between the cardiovascular system and your energy, so how much carbohydrates did you start with, how many lipids did you have, how much mitochondria do you have, this is what limits what the muscle can do. And this is also why, as somebody exercises, the muscles tend to grow more mitochondria. And so with practice, the person who exercises a lot will generally not fatigue as easily because they have more the mitochondria to produce the energy, versus somebody who doesn't bolt that often and they suddenly start, they'll only have a few mitochondria and they won't be able to sustain them very long. Okay, the very last thing for this lecture is just a comment on temperature. So exercise creates heat. Right? We said that this is useful when we're shivering and you're trying to warm up. You release extra heat. But as you're exercising for the purpose of getting somewhere, making extra heat is probably not your objective. This becomes an annoying side effect. And it's annoying for a few reasons. One is because as your core temperature does rise, you will now start engaging in the cooling off system, the cooling off programs, such as sweating. So you're exercising and you're sweating. Okay, not too much of a problem, except that as you're sweating, you may be losing electrolytes, maybe losing water, so you could be dehydrating yourself in the process. And with the electrolytes comes the risk of having muscle cramps. Also, reduced blood volume. The second part is that you divert the blood flow towards the extremities, towards the skin. Right? That was the point, so that as the warm blood reaches the skin, it can cool off. So now we have a competition. Your muscle wants the blood flow, your skin wants the blood flow, your brain wants the blood flow. That's a lot of kind of people to appease. And that's where there is the potential that the person will pass out because there's competing interests and the blood will end up going to the periphery, to the cutaneous system, and maybe not enough of it will reach the brain and the person will start to feel faint or pass out, depending on the degree of how much they're overheating. So that will dictate how much of a priority it is that the blood go to the cutaneous system as opposed to the brain. Acclimatization means that over time, you generally adapt to feeling hot and exercising, and you tend to optimize these pathways better. So a person who exercises a lot, they might start to sweat earlier to keep their temperature lower sooner. And the person who doesn't exercise as much, it's almost like their body doesn't know it's coming, so it doesn't plan for it as well. As somebody who does it on a regular basis, then their body kind of adapts and understands that this is efficient, this is an effective way to keep the temperature going. This is pretty much it for today, for this lecture. So before we end, we are ending early, but I wanted to go through the midterm in some form. The midterm and a little bit of review, just the quiz questions that maybe came up. So first of all, the exam is multiple choice and short answer. It is 50-50, so 30 multiple choice questions and 30 marks coming from the short answer. To preempt, I see a very even distribution of marks in the sense that there are people who do equally well or equally poorly on both. So they're either succeeding well on multiple choice and short answer or tanking equally on both. And then in the middle group, you'll have people completely on polar opposites. where somebody who does super well on multiple choice may tank on the short answer, and vice versa. People who really love the short answer tank on multiple choice. I say this now because over the years I've heard both sides who tell me that I should switch the format because the format doesn't work for them. And I understand that. It's just that in a group of 100 people, there are other people for whom it does actually work this way. And as odd as it is, it does actually flip pretty evenly, 50-50 in terms of who does well with the writing part, who does well with the multiple choice. It is 90 minutes. So this part is actually really important. 90 minutes means that 30 multiple choice questions should take you, actually a second. So we have a 1.2 ratio. So 1.2 times 30. It should take you 36 minutes. So that's really important. So maybe put a timer on, or bring a watch, or just gauge it wherever you are, because that means that if we take 90 and we subtract 36, we're left with 54 minutes. So 54 minutes should be dedicated towards the short answer. So there are 6 questions, so that is 9 minutes per question. I also emphasize this because we also get into the conversation that why run out of time, and if only I had more time for the short answer, it would work. And when I ask how long would that be, well, between 6 and 9 minutes. Like, okay, but we actually have 9 minutes. And I agree, it is a very challenging thing to do, to stick to the 1.2 for the multiple choice. So maybe switch it around for yourself. Maybe start with the short answer, prioritize that, give yourself 50 minutes for the short answer, and then the rest of the time for the multiple choice. Because if you do multiple choice first, it does take a lot of discipline to cut yourself off and be like, okay, I'm out of time, I have to move on to the short answer. Okay, content, it's everything up until and including today and this Friday. So fever, the coagulation, the cell physiology, anemia, nerve, and muscle, and then the same for labs. And now we'll go through a few questions. Which of the following would not potentially result in thrombocytopenia or reduced platelet count? Which would not result in this? I think this was already a multiple choice quiz. Yes? Okay. So this would be thrombopoietin, and I will be posting these, so that's good. I just need to unhide them. So thrombopoietin is that TPO, which is made by the liver, which promotes the production of platelets in the bone marrow. So thrombocytopenia is a reduced count. So if you had too much TPO, if anything, you'd have too many platelets. Elevated mean platelet volume can be seen in all of the following except. So mean platelet volume is how big is the platelet, right? And we tend to see larger platelets. These are newer platelets. So TPO deficiency, yes, because that means that we wouldn't be making new platelets. If anything, they would be small because with age they would shrink. Intrinsic and extrinsic pathways both function to activate which enzyme? So maybe it's a sneaky question. So thrombin does act as an enzyme by converting fibrinogen into fibrin, right? And that's where the common pathway meets, where we have factor 10 that activates the thrombin, and then thrombin is able to convert fibrinogen into fibrin. Hemolytic anemia. So this is the part where we destroy red blood cells. We have many different ways of doing the destruction. It could be done by parasites. Our own immune system can do it. Or if there is something wrong with the cell itself, for example, sickle cell anemia, where the hemoglobin is different, then the cell can be self-destroyed. Sorry, it can be destroyed by itself because it'll rupture because of its malformation. The key feature with hemolytic anemia is that usually there is an increased RDW because there is an increase in production of red blood cells, but also because what is left looks very different. Typically, a sickle cell anemia, even the cells that are remaining, they look different shapes. You have sickle shaped cells and you have healthy ones. So because of that, you have a very large variability in terms of what the cells look like on average. You have too many different looking cells. Mean corpuscular volume. Lack of hemoglobin leads to small size. Right? I highlight this because you'll see questions, especially on the short answer, that will ask you to interpret a case. Very similar to the cases that you've seen in the anemia lab, but the questions will be, can you please explain why they look the way they do? So not just iron deficiency anemia, but like why? Who cares? Why? So the iron is less. Why does it change the cell size? If you have less iron, your I don't know, skeletal cell isn't going to shrink necessarily. Why this one? And the reason is that hemoglobin is making up pretty much the entire volume of the cell. And hemoglobin is dependent on iron. Don't have the iron, can't make the hemoglobin. And without it, you'll make a smaller cell. Whatever amount of hemoglobin you have, okay, you'll make half the cell that you usually would. Why are the cells large in B12 and folate deficiency? The reason is that you need both of these nutrients for proper DNA maturation and for the cells to properly divide. Otherwise, they get stuck. So we have our mitosis phases, and the S phase is where you basically just doubled your chromosomes. You're about to start double checking them before you separate the two cells. And if you get stuck here and you go to production like this, you'll end up with extra chromosomes, your bigger cell. You were supposed to divide, but you never did, so you'll be huge. High transferrin iron binding capacity is suggestive of what? D. So iron deficiency means that you have very little iron, so your transferrin can bind lots of it. It's empty. It has lots of carrying capacity, which just got no iron to bind it. So transferrin iron binding capacity is very high. My hands are empty, I can carry a lot. So iron deficiency for that reason, because there's very little iron to actually go around. Low ferritin maybe is a little bit sneakier. So ferritin, we said, was a storage molecule. But the term ferritin itself implies that it has the iron with it. So if somebody has low ferritin, it means that they have low iron as well. So that effectively means that C ends up being B. Question?
Would there ever be ferritin that wasn't bound iron?
Is it possible? There are cases where ferritin is very, very high, where we have a person suffering from inflammation. But in that case, is it truly that their ferritin is empty, or did they just hoard all the iron and stored it in the ferritin? I think that it always does have iron bound to it. Again, it's one of those where I would need to, I'm like 98% sure, but ferritin without iron. Okay. If the patient presents with macrocytic hypochromic anemia, which stage of erythropoiesis is most likely to be affected? It is also a sneaky question, and this one was dependent on your article. So the article talks about, this was more so with the B12 and the folate, that as you affected the DNA synthesis and you couldn't properly finish that chromosomal divide, you were actually unable to complete the hemoglobin production either. And from a concept perspective, it kind of makes sense in the sense that if your DNA isn't properly edited and you haven't actually divided it properly, everything else that depends on the DNA is also going to be crappy. In this case, it's the hemoglobin part. Elevated RDW is likely to occur in which of the following situations? So this one would have been changed, actually. Sorry. This should say, and all of the above. And this is cool because a year ago, all of the above, my understanding of this was that it was only A and C. And a year ago, somebody on the team in the lab discovered an article that iron deficiency also correlates with increased RDW. And for that reason, now we have all of the above. So live and learn. The article doesn't expand on kind of why specifically iron deficiency and not necessarily B12 or folate deficiency doesn't fit into this. But it appears that as you're hitting anemia of this type, you're stimulating increased production, maybe to make up for it. They're kind of saying, you know what, they're small, they're not really functioning properly, let's just make a lot of them and hope for the best, that at least the numbers will make up for it. So generally, increased reticulocyte production, this is happening when you feel like you don't have enough oxygen, you don't have enough red blood cells to carry it. Which could be because you were hemorrhaging, because there was hemolytic anemia, or because you have the iron deficiency anemia. All of the following are correct about thromboplastin except. Oops. B is correct. So we have thromboplastin, its other name is tissue factor. And tissue factor resides within the endothelium and outside of it in the tissue. So that's why it ends up activating the extrinsic pathway. And it is definitely pro-thrombogenic. So it is located within the endothelium. It does activate clotting factors, and it's definitely an extrinsic pathway activator. I think there's just maybe one more and then short answer question. Another most likely consequence of the overactive spleen that destroys erythrocytes at a faster rate would be what? Jaundice. Okay, that's cool. So we don't even all include this, but essentially, why jaundice? Delivery, bilirubin. Yeah, exactly. So the iron we can restore, where we can recycle. The proteins, the hemoglobin, the globulin, we can recycle. But the heme, we have to get rid of. The heme component becomes bilirubin, and if you can't get rid of the bilirubin at a fast enough rate, it'll turn to jaundice. So if you have somebody who has the spleen that is destroying things very quickly, then the other organs like the liver and the kidney may not be able to keep up with clearing the bilirubin. And for a while, the person may appear yellow until they clear it. What type of anemia would this be if you have an overactive, spleen that is actively destroying red blood cells? Hemolytic anemia. Nice. Okay, almost there. Patient presents with new onset dyspnea, which is shortness of breath and fatigue. Past medical history reveals a new medication that started 2 weeks ago. His CBC is ordered, and it's unremarkable except for increased RDW, increased elevated MCV, low hematocrit, and low red blood cell. So what's the name for this presentation? Macrocytic and normochromic. Yes. So we know that the only problem was the increase in size, macrocytic, and we were told that everything else was fine, so it would be normochromic. Okay, finally, short answer question. If this patient's MCH was reduced, so actually let's do a brand new person. We have a person and their MCH is reduced. What would this suggest about their anemia? So let's say the only thing that you got back for whatever silly reason is just their MCH. Please explain how you would interpret this. So provide your rationale and please comment on the erythropoiesis and how it is causing the anemia. So this is pretty much actually provide your rationale. This is a copy and paste from the exam. This portion will vary, the beginning part, and the case will vary. But the provide your rationale is the same, and the erythropoiesis. So I'm gonna go through and break it down how to answer questions like this. So this is one way to think of short answer questions in general. The first part is to get the fastest response out there so that if you have very little time, you at least give a direct response and you could move on. But typically a direct response buys you very few marks. The questions are all 5 marks, so we'll use that model. If you can't provide an example, sometimes this won't make sense. Otherwise, make sure you've talked about the pathways that lead up to whatever this is. So if we're saying, talk about erythropoiesis, that's the pathway. Mention key structures and key hormones, or key hormones. So maybe you're talking about erythropoietin in this case. Macrocytic, right, we said, was reduced, so this is hypochromic. So hypochromic means hemoglobin, so that's the structure we'll talk about. Maybe you can talk about iron. what's the net effect, and then relate it back, find a way to close. So this is basically the marking rubric that you would see if you choose to go and review your exam, which, by the way, highly recommended. There's a step 2 that happens soon after you do your exam. This is actually important. Did anybody talk to you about step 2, step 3, what they are, who they are? Yes?
Yeah, it was taught today in the town hall meeting.
Mm-hmm.
And so, I mean, that when we send out the email, we'll kind of
Summarize it.
elaborate about that.
Thank you.
Yeah.
Okay. I'll talk about it anyway.
Yeah.
Just because it is important. So basically, after you write your exam, there is a period of time when you get your results back where you can sign up to see your exam. The sign up is usually, let's play the worst case scenario, it's hidden somewhere far, far away and nobody ever knows where it is. So you can already start to think about how you're going to contact your class rep, who you're going to ask to find out where this is. I know that every year we send out, there's emails, there's highlights, it's somewhere in Moodle, and every year somebody loses where it is. So there is a mysterious place where you can sign up for a step 2, probably somewhere in Moodle, and there's a deadline. And the deadline is usually like 48 hours from the time that you see your exam. So it's very short, and so kind of be ready. There's no cost to you seeing your exam, right? You can pretty much see all of your exams. If it's a short answer question, definitely come and see your exam, because that way you'll get to see the marking rubric, the thought behind these questions. If you're getting a great mark, okay, you don't have to, but you can still come and check it out. If you didn't get the mark you wanted, definitely come and check it out, because sometimes it's just a question of how much more detail we want versus what you were expecting. Most of the time, that's where the negotiation is, or the mis-expectation. You have 2 steps. Step 2, it means that you're meeting with an invigilator in a closed room, whatever, with a group of your colleagues, and you're all gonna review your own personal exams with the answer key. You can go through your multiple choice, you can go through the short answer, and you can compare and contrast and figure out what it is that you want to do. Step 3 is where we meet individually. And